we'll have time to get to the downsides of the way they scaled up their production on these things. But if we can separate the marvel of what they did, yes, there's an awful lot of stuff here that's beyond wizardry. It's just incredible what they accomplished.
I'm sorry I don't want to take you off track, but you were describing what would happen if it went to various organs that would damage them. Could it cause cancers too?
We can get back to that. We clearly are seeing an uptick in cancers, and an uptick in cancers that are unusual, especially in their speed. Maybe if we have time, we can come back to the reasons that might be occurring. There's a lot of discussion amongst the medical dissidents about why that pattern exists and what it implies. But yes, clearly cancers are one of the failure modes of the body, and this highly novel technology clearly had that as a risk, even if we didn't know what mechanism it would happen by. But yes, let's say you're a soccer player and you've been injected with this stuff, and a bolus of it has hit your heart and caused a bunch of your cells to be destroyed by your own immune system, by cytotoxic T cells and natural killer cells. Well, now you've got a wound. If you manage to survive to have it scar over, then that wound will be less of a vulnerability than it would otherwise be. But if in the period after you've been damaged, before your heart has fully scarred, you were to push yourself to some new athletic limit, let's say you're in the middle of a particularly intense game, right? That would be exactly the time when a weakness in a vessel wall might cause a critical failure, and you could die on the field. So this was a very plausible mechanism to explain the pattern of sudden deaths that we have seen oftentimes in people who are unusually healthy and athletic.
Yes.
So to go back to the original story, pharma had a potentially tremendously lucrative property that it couldn't bring to market because a safety test would have revealed this unsolvable problem at its heart. And so what I'm wondering, my hypothesis, is that it recognized that the thing that would bypass that obstacle was an emergency that caused the public to demand a remedy to allow them to go back to work and to living their lives, that would cause the government to streamline the safety testing process so that it wouldn't spot these things. And indeed, one of the things that we see, in addition to a lot more harm in those safety tests than we were initially allowed to understand, but also the safety testing was radically truncated so that long-term harms were impossible to detect. So the hypothesis in question is pharma used an emergency to bypass an obstacle to bring an incredibly lucrative technology to normalize it in the public, in the regulatory apparatus, to sneak it by the things that would ordinarily prevent a dangerous technology like this one from being widely deployed.
So I think that sounds...
Howard Martin Neman observed the phenomenon of reverse transcription, and David Baltimore discovered the enzyme reverse transcriptase, which is involved in the process of reverse transcription. The enzyme reverse transcriptase is an RNA-dependent DNA polymerase, which means that it will seek RNA as a substrate and transcribe or reverse transcribe it into the DNA. Which means that it will seek RNA as a substrate and transcribe or reverse transcribe it into the DNA.
Which means that it will seek RNA as a substrate and transcribe or reverse transcribe it into the DNA.
Which means that it will seek RNA as a substrate and transcribe or reverse transcribe it into the DNA fragment. The process of reverse transcription involves the following steps: annealing of the primer to the RNA transcript or to the poly A tail in case of eukaryotic messenger RNA, reverse transcription by reverse transcriptase, removal of mismatches by RNase H, repair of gaps by DNA polymerase I cleanome fragment, and finally, the complementary DNA strand, termed as cDNA, is ready for downstream processing. Enzymes employed for reverse transcription in the laboratory are reverse transcriptase, RNase H, DNA polymerase I cleanome fragment. The process of reverse transcription begins with annealing of the primer. RNA tends to form secondary structures via Watson-Crick base pairing. The first step involves denaturation of the RNA by heating in the presence of the gene-specific primer, followed by cooling to room temperature to facilitate primer annealing. The blue represents the RNA strand. The yellow represents the primer which has annealed to the RNA strand. Now, in the case of eukaryotic messenger RNA or mRNA, the primer which can be utilized is an oligo-DT primer which binds specifically to the polyadenylation signal at the end of the messenger RNA sequence. In the case of prokaryotic RNA, the primer which needs to be utilized is the gene-specific primer. The second step involves reverse transcription, which is facilitated by the enzyme reverse transcriptase. Reverse transcriptase commences transcription at the primer binding site. In the slide, the reverse transcriptase is depicted in black. Following the activity of reverse transcriptase, a double-stranded RNA-DNA hybrid is generated. This double-stranded DNA-RNA hybrid needs to be repaired in order to synthesize cDNA. The enzyme RNase H is involved in the removal of mismatches. The mismatches in the DNA-RNA hybrid generated by reverse transcriptase are removed by the enzyme RNase H. Finally, step 4 involves the repair of strand by DNA polymerase I cleanome fragment. DNA polymerase I cleanome fragment repairs the gaps and fills in nucleotides, leading to the formation of a double-stranded DNA. The resulting DNA strand can be subjected to DNA sequencing in order to
mRNA is the key ingredient in vaccines helping us fight COVID-19. But what is mRNA? Here, in the human body, our cells are constantly making proteins to keep everything running smoothly. mRNA, or messenger RNA, is a key part of this process. It carries the instructions, or code, from the DNA to create different proteins in the cells. In a vaccine, mRNA is delivered inside a protective fatty coating, which can get into our cells to release the mRNA code. It instructs the cell to create a spike protein, the same protein you'll find in the COVID-19 virus. The body recognises these new spike proteins as foreign and then learns how to identify and fight the real virus. mRNA vaccines are only the beginning of a revolution in medical technology based on RNA. The possibilities for applying RNA research across a range of areas, such as cancer, rare genetic disorders, and infectious diseases, hold huge potential.
In a moment, the proviral DNA is copied by cellular DNA-dependent RNA polymerase to make mRNA, and these mRNAs go out into the cytoplasm by the normal cellular pathways. They can be translated to make glycoproteins, the envelope glycoproteins, which go to the plasma membrane. They can be translated to form the capsid proteins, the GAG proteins, and the polymerase. or they can go into new virions. It's all the same messenger RNA with the exception of that one that's spliced. If these were all, this one is a spliced mRNA because it encodes the envelope proteins, but these two are full length and this one goes into virions. So the genome of a retrovirus is nothing more than an mRNA that is made by transcription of an integrated copy of the proviral DNA. And new viruses are then made by budding, which we will talk about later on. Now, reverse transcriptase is an enzyme that copies RNA to make DNA. All the same rules apply as for other enzymes that we've talked about. The template is copied in a 3' to 5' direction. The newly synthesized nucleic acid is made in a 5' to 3' direction by stepwise addition of triphosphates to the 3' end. And reverse transcriptases require a primer. They are a primer-dependent enzyme. The primer can be RNA or DNA. Doesn't matter. The primer must have a paired 3' terminus, so this base here at the primer, the last base, has to be base paired. It can't be floating away. Some enzymes don't mind if the 3' end is not base paired, but this one does. The template can be RNA, which is the viral RNA, or it can be DNA. You'll see why that is important. And deoxy-NTPs are incorporated, not RNA. It does not make RNA, this enzyme. It makes just DNA. The template can be DNA or RNA, but it only makes DNA. And, of course, just like every other polymerase, the incoming deoxy-NTP is selected by base pairing here. So that's reverse transcriptase. Now, reverse transcriptase has associated with it a second activity, which is a separate domain in the protein. It's an enzyme called RNase H. So this is an enzyme that degrades RNA, as you can gather from the name, but it has a very specific activity. First of all, it will cleave RNA. It's an RNase, but it only does so when the substrate is double-stranded or in duplex form. That duplex can be RNA-RNA, it can be double-stranded RNA, or it can be RNA-DNA. Either one, doesn't matter. The RNase will cut the RNA strand. It's an endonuclease, which means it cuts internally. It doesn't chop the ends off, which would be an exonuclease. So enzymes that take off one base at a time from the ends, those are exos. This is an endonuclease. It cleaves internally. And it makes short pieces of RNA, oligonucleotides, with a 5' phosphate and a 3' hydroxyl. So here is a duplex molecule, could be DNA, RNA, or RNA, RNA. RNA.
Joseph Freeman and his colleagues, including Peter Doshi, did an evaluation of Pfizer's own safety data from its safety trials. Now, these trials were absurdly short. Pfizer only allowed 1 month before it vaccinated its controls and made it impossible to detect further harms. And what they found was a 1 in 800 rate of serious adverse event. This is not minor stuff. This is serious harm to health. 1 in 800 per shot. That's not per person. That's per shot. 1 in 800 rate, which in 1 month, that suggests a very high mortality risk. And in fact, we saw mortality in the safety trials. What happens over the long term? We've certainly seen such a range of pathologies that have crippling effects on people's health that I shudder to think how many people have actually.
So I'm not a math genius, but 1 in 800 shots times billions is, a lot of people.
Yep. There was I was recently at a conference in Romania on the COVID crisis, and so there was a lot of work trying to unpack what we actually understand, and I saw a credible estimate of something like 17 million deaths globally from this technology. So
17 million deaths from the COVID vax?
Well, when you scale up to billions, it's not hard to reach a number like that with a technology this dangerous. Now, to your deeper question, I think let's steel man.
So just for perspective, that's like the death toll of a global war.
Yes, absolutely. This is a great tragedy of history. So that proportion.
Then took the vaccine, the Pfizer-BioNTech, they diluted that vaccine with 0.9% NaCl. They brought it to the exact concentration that they wanted to test with, and they created 0.5 milligram per milliliter, 1.1 microgram, not milligram, 1 microgram per milliliter, 2 microgram per milliliter. And then they put those various concentrations on various wells. They incubated this whole thing, these cells, with or without vaccine. So on some cells, they had put the vaccine. Some cells did not have the vaccine. Those cells that had the vaccine, over there was 0.5 or 1 or 2. Then they kept those cells in the presence of the vaccine or absence of the vaccine as a control for comparison for 6 hours, 24 hours, and 48 hours. So what are they trying to figure out? What they were trying to figure out is that if they put this vaccine on the cells, what will happen to the vaccine? What will it do inside the cells? What will it do in 6 hours? What will it do in 24 hours? What will it do in 48 hours? And they also had a control where the vaccine was not in the cells. So they would say that if the cells are sitting by themselves without any vaccine, what is happening in them, so that they can compare the activities and understand what activities were because of vaccine, because of the concentration of the vaccine, and because of the time versus not vaccine. So that is this part. This part. So if you see here, HUH7 cells were incubated with or without BioNTech vaccine. Then is a real-time qRT-PCR. After this, when the cells sat with the vaccine, their question is, in their head, will this vaccine enter the cells? If so, how efficiently? One. Second, once inside the cell, what will this vaccine do? Will it create DNA? If so, how much? How fast? So these are the kind of questions that they're trying to answer. So if you see here, the next step. So after 6 hours or 24 hours or 48 hours and various concentrations, they opened up the cells, right? And they extracted the material from the cell. They extracted RNA, they extracted DNA, and they extracted a very specific RNA for the cell, which is called line 1 RNA. And because today we are doing a discussion which is in detail, why were they interested in line 1? Line 1 is a special protein which is present usually in dividing cells. And remember, these are human hepatoma 7 cells. These are cancer cells. So they are by nature dividing. In us normal people who do not have cancers or in non-cancerous cells, our body is composed of 2 types of cells at the bigger level, division level. Those cells that are dividing, for example, cells in the bone marrow are dividing, immune system cells are being produced, blood cells are being produced. So these are dividing cells. Then cells that are dividing in the GIT, our GIT surfaces are always getting replenished. Cells of the skin, even the respiratory system cells, and so on. You can actually, sometimes when you see somebody going through chemo and you see the kind of issues that develop, they get immune suppression because the chemo would suppress dividing cells. So because immune cells are generated by division, that immune suppression occurs. They get GIT problems because GIT cells are replenished all the time. And when the chemo stops the GIT or reduces the division of the GIT cells, because chemo is trying to reduce the division of the cancer cells, it would just reduce division in all cells. So the patient would develop GIT issues. Similarly, they would develop skin issues. That is why they lose hair, because the skin cells are not replenishing correctly as well. And similarly, they'll develop other issues. So same thing over here. These are hepatoma cells. These are dividing cells. Now, inside these cells, there is a special protein called line 1. Line 1 protein has a special role, and that is it helps with the reverse transcription. Reverse transcription means to take an RNA and convert that into a DNA. There are some viruses that actually have their own enzyme that can take their RNA and convert that into DNA. Here we are looking at a human protein that can take up possibly SARS-CoV-2's RNA or this vaccine's RNA. This paper is about the vaccine, not the SARS-CoV-2. So take up the vaccine's RNA and convert that into DNA. So the question is, will it do that? So to answer that question, researchers are first going to see, will the vaccine cause an increase in line 1? If it is going to cause an increase in line 1, that means there is a possibility that when there is more line 1, that might cause these DNA creation as well. So here, what they did was, they took those cells and they extracted the RNA. There were two types of RNA they were really interested in. One were the RNA of the vaccine itself, and the second one were the RNA for the line 1. So here you can stop and say, well, we are talking about DNAs. All of a sudden now we are talking about RNA for line 1. What is this mess? So it is actually very simple. To create the DNA, we need the line 1, for example. Line 1 is a protein. To generate line 1, we need the recipe of the line 1, which is the DNA gene. So let's say here, this is a nucleus. Inside the nucleus, imagine that there is a gene for line 1. We need to express that gene. We need to create messenger RNA from that gene. Remember, it's a recipe. So when we take the recipe book, that is DNA's genes, and we make a copy of that recipe, that is RNA, then we bring that RNA outside of the nucleus, inside the cytoplasm, cytosol as well. We have ribosomes that will connect with this RNA and make the protein. So if we want to make line 1 protein, we have to first open up the genes for line 1. We have to then make a copy of the recipe in the form of messenger RNA for line 1. That messenger RNA should come out from nucleus into the cytoplasm. In the cytoplasm, it should connect with ribosome, and line 1 protein should be formed.
One, two. One, two. One, two. That's how I roll. I have one speed, I have one gear. Your anchor. One, two. Okay, your passion is coming off as erratic. Your anchor Okay, your passion is coming off as erratic. One, two. One, two. One, two.